Hi, I am Mehar Jitendra Mehar. I registered my PhD at AcSIR under the discipline of Biological Sciences, and I carried out my PhD work at CSIR-CFTRI Mysore. My PhD thesis is spirulina pratensis cultivation using automated open recipe pond with CO2 fed pH control, metabolite profiling and microbial population dynamics investigation. Spirulina is a type of algae that has potential benefits in addressing several sustainable goals, as it is a rich source of protein, essential amino acids, vitamins and minerals, and can be used as a nutritional supplement or food source to combat malnutrition and food security, which aligns with Sustainable Development Goal 2, Zero Hunger. Additionally, spirulina has the ability to sequester carbon dioxide during its growth and can be cultivated in areas where traditional agriculture might not be viable, helping to reduce pressures on land use, which aligns with SDG 13 climate action. Since 1970, commercial production of spirulina has been carried out at moderate to large scale because of its high market value, process sustainability, and easy value chain. Conventionally, spirulina cultivation is carried out using Xerox medium without pH control, wherein sodium bicarbonate is added as 16.8 gram per liter as a carbon source. However, pH is one of the most important factors for cell growth, controlling microbial contamination as well as CO2 mass transfer in the cultivation medium. In this study, an indigenously developed automated system for spirulina cultivation was used at pilot scale, where CO2 fed pH control was aimed at providing a carbon source by replacing sodium bicarbonate and to control the culture pH. Amongst all the studied CO2 fed pH conditions, that is pH 7.5, pH 8.5 and pH 9.5, pH 8.5 was found optimal for biomass productivity, 72 mg per liter per day, CO2 biofixation rate 0.13 gram per liter per day, protein 64% and phycocyanin 14%, along with lower ash content of 7% compared to the conventional ash content of 12 to 15%. The pilot scale automation strategy developed in this work can be employed to enhance the performance of the spirulina or any microalgae cultivation. C-phycocyanin is a major phycobiliprotein representing up to 14 to 20% of the spirulina. C-phycocyanin has gained significant attention in nutraceutical and pharmaceutical industries. The application of C-phycocyanin varies based on the degree of purity. A purity more than 0.7 required for food, more than 1.5 for cosmetics, up to 3.9 for reagents, and more than 4 for pharmaceuticals and analytics. Hence, developing efficient, sustainable, and economic purification strategies with minimum steps are crucial for commercial application. In another objective of this doctoral work, comprehensive and integrated multi-step purification designs were conceptualized by combining protein purification techniques such as ammonium sulfate precipitation, membrane filtration, with an intermediate clarification step. A purification process designed for resulted in purity of 3.15, purification fold of 4.46, and recovery of 81.59%, which is acceptable to be used in food and cosmetics. To overcome fouling, membrane cleaning using hydrogen peroxide of 50 millimolar resulted in relatively higher cleaning efficiency of 94.08% as compared to ozone, which is 57.51%, and phosphate buffer, which is 51.43%. These findings emphasize effective sustainable methods for purifying crude phycocyanin from natural sources. Using next generation sequencing methods, This study also aimed to investigate microbial community composition and functional potential of spirulina cultures cultivated at conditions of CO2 fed pH control at pH 8.5 and pH 9.5, and the results were compared with the conventional Xerox medium. A total of 53 high quality draft genomes of taxa belonging to phyla Proteobacteria and Actinobacteria were retrieved by genomic binning. However, their abundance was relatively higher in ZM as compared to CO2 fed pH control conditions of pH 8.5 and 9.5. This thesis aimed at achieving cleaner and sustainable spirulina production and phycocyanin purification. Production costs associated with sodium bicarbonate can be saved. Only initial capex for adding the CO2 based pH control is required. जय हिंद, जय भारत.